Life cycle of marsilea. Classification, division, pteridophyte, class Phelicopsida, order Hydropteris, family Marsilaceae, genus Marsilea. Marsilea is commonly known as water fern. It is the aquatic and semi-aquatic plant in the temperate region. Plant body is sporophytic and herbaceous plant. The plant body differentiated into rhizome, leaves and roots. Reproduction, sporophytic space. Spores producing organs. Marsilea is heterosporous. It produces the two types of spores. It's a microspore and megaspore. The spores are produced in the microsporangia and megasporangia. The sporangia are born special type of spore producing organ called sporocarp. The sporocarp are born laterally on the shortened lateral branches of the petiole of the leaf, either near the base or a little higher up. The array is solitary or in clusters. Number of sporocarp differs in different species and varies from 1 to 20 or more. Each sporocarp is an oval and bean shaped. It is the flattened structure. It is green and soft when it is young, but at maturity it becomes very hard and brown in color. Development of sporangia, and the development of micro and megasporangia of leptosporangiate type. The leptosporangia differ from the other ferns having a sporangium with jacket layer 1 cell in thickness. There is a defined number of spores within sporangia. The sporangium develops from a single initial cell and tapetum differentiates from a single initial cell of developing sporangium. And this is the development of micro and megasporangium. They divided into the two cells, apical cells, and the primary jacket cell will produce, and then jacket cells are grow, then tapetum cells are grow. Next, sporogenous tissues are grow. Then micro and megasporangium will be produced in the following steps. Dehiscence of sporocarp and liberation of spores. The decaying of the wall of the sporocarp takes place due to the bacterial action, and the sporangia and spores liberate. The sporocarp bursts open only in water. The gelatinous ring absorb water and extend greedily through the open margin of the sporocarp, thus dragging out sori along with it. The gelatinous ring bears two alternation rows of sori. The delicate mucilage wall of the sporangia open in water, and the spores are liberated, which germinate soon. This picture clearly explains the sori development. Gametophytic phase. The microspore and megaspore are the unit of male and female gametophyte. Microspore is the smaller, megaspore is the larger one. Microspore is the male, megaspore is the female. Development of male gametophyte. Microspore divided into two cells. The smaller became prothalae, and it reduced the male gametophyte. The larger divided into the antherial cells. The antherial divided into three jacket cells and single archegonial initial. The archegonial initial of each antherium divided into produces 16 anthrocytes, each in the anthrozoids. Anthrocytes have many coils and flagella, and the other prothalae cell and jacket cells are disintegrate, become free in the surrounded water. Development of female gametophyte. Megaspore divided into two cells. The smaller cell occupies the whole peak. The larger cell does not divide in further. The smaller cell function as an apical cell. The apical cell function as an archegonial initial. Archegonial initial divided to produce a small primary cover cell at the top, at the central cell at the base. The cover cell divided from four neck canal initials. They divide to form a neck. The central cell divided to produce a small upper primary canal cell and lower larger primary ventral cell. The primary canal cell divided to produce two neck canal cells. The primary ventral cell divided to produce a large lower oospose, it's the egg, and an upper smaller ventral canal cell. The ventral canal cell and the neck canal cell of the mature archegonium disintegrate. It forms an opening for the entry of anthrozoids. This is the male and female gametophyte development. Fertilization. Many anthrozoids are attached towards the funnel-shaped structure of the megagametophyte. The anthrozoids enter gelatinous envelope and reach to the archegonium. Only one anthrozoid enter into the egg cytoplasm and fuses with the egg nucleus to form the zygote. Development of embryo into the new sporophyte. The zygote is mother cell of the next sporophytic generation. The first division of the zygote is vertical, followed by the transverse division, resulting in the coordinate stage of the embryo. Subsequent development of the upper two cells from a root and leaf, whereas the lower cells give rise to foot and shoot apex. This is the alternation of generation and life cycle of marsilea. And the marsilea plant, it have a sporocarp. The sporocarp divided into the microsporangia and megasporangia. In the micro and megasporangia will produce the microspore mother cells and megaspore mother cells. Under the meiosis division, the microspore will produce the microspore, megaspore mother cell will produce the megaspore. The microspore to become a male gametophyte, the megaspore to become a female gametophyte. It produces the male gametophyte, produces the antheridium, female gametophyte, it produces the archegonium. Then next to the step, the antheridium to modify the egg, and the antheridium to produce the antherozoids, and the archegonium will produce the egg. Then fertilization will happen, then zygote form and embryo form. This is the life cycle of marsilea. Thank you so much.